So from now on, we are going to discuss about the tumors of the connective tissues or the mesenchymal tumors. So mesenchymal tumors are tumors arising from the mesenchymal tissues like fibrous tissue, adipose tissue, muscle tissue like smooth muscles and skeletal muscles, cartilages, bone and blood vessels, and also from nerve fibers. So these tumors can be classified into basically into two types like benign tumors and malignant tumors. So, the benign tumors are so when talking about the tissues and the benign tumors and malignant tumors which they make, here are the summarized table of it. So from fibrous tissue, they can make a benign tumors called fibroma and malignant tumors called fibrosarcoma. So from adipose tissue, they can form benign tumors called lipoma and malignant tissues called liposarcoma. So as you can see when you read in these, the benign tumors are often ended by the word oma. Look here, fibroma, lipoma, chondroma, osteoma, leiomyoma, rhabdomyoma, hemangioma. But when ending the malignant tumors, they are ended as sarcoma, fibrosarcoma, liposarcoma, chondrosarcoma, osteosarcoma, leiomyosarcoma, rhabdomyosarcoma, angiosarcoma, and synovial sarcoma. So, when discussing the differences between benign connective tissue tumors and the malignant connective tissue tumors or the sarcomas, benign connective tissue tumors are well circumscribed and they are encapsulated. But the malignant connective tissue tumors are poorly circumscribed and they infiltrate. So, benign connective tissue tumors are slow growing while the malignant ones are rapidly growing. The benign tumors resemble with the tissue of origin, so they are well differentiated, while the malignant ones are anaplastic or poor differentiated. Benign tumors, there are no metastasis or there is only a minimal mitotic activity, while the malignant connective tissue tumors has frequent mitosis and hematogenesis spread to lungs or metastasis. So let us discuss about each and every connective tissue tumors. First, we will discuss about the peripheral nerve sheath tumors or the PNSTS. So tumors arising from the components of the peripheral nerve, such as from the Schwann cells, perineurial cells, and fibroblasts, are called the peripheral nerve sheath tumors. So they can be also classified as we discussed earlier into two as benign and malignant. So the benign ones are the schwannomas, fibroma, neurofibroma, and perineurioma. And the malignant ones are the malignant peripheral nerve sheath tumors. When discussing about the schwannoma, or we call it also as neurolemoma, and there are key features of a schwannoma. They are benign. So because they are benign, they are encapsulated tumors. And as its name says, it is made out of Schwann cells, and they grow eccentrically from the nerve. And the common sites where you can find schwannomas, the vestibular nerve or the eighth cranial nerve. Actually, most of the time it is commonly seen at the cerebropontine angle where the vestibular nerve comes out from the intracranium. And these vestibular nerve schwannomas, we also call it that acoustic neuroma. So, uh, uh, okay. Now let us discuss about the histology of these schwannoma. So focus on this slide. In this slide, you can see an dark area and a light area. So this dark area, which is hypercellulated and stained well, is known as Anthony A areas. And these areas, which has a low number of cells and which has a loosely arranged myxoid stroma, is called an Anthony B area. So these two areas are seen in schwannoma. And also another thing you can see in an histological side of an schwannoma is verrucate bodies. Verrucate bodies is complex like this, that there are two palisading nuclear rows, and in between these nuclear palisade there is an anuclear zone where there is no nucleus. So these are together known as varicobodies. And this is a actual slide where you can see these varicobodies. Look at inside this circle. So you can see here there is a varicobody. Also in the outside you can see there are varicae bodies like this. So what is the clinical importance of knowing this schwannoma? So, as I said earlier, the most common site where these schwannomas occur is the eighth cranial nerve. It is the vestibular nerve. So, the patient might get hearing loss or tinnitus, and also this is associated with neurofibromatosis type 2. When discussing about the gross features of the schwannoma or gross features of the microscopic features, you can see this is well circumscribed and encapsulated. So look at this. This is well encapsulated and it is oval and a spherical mass and eccentric to the nerve. It means it pushes the nerve aside and does not infiltrate into it. So look at this picture A. Focus on this picture A. So you can see this is the nerve and this is the schwannoma. Instead of infiltrating to the nerve, it has pushed the nerve away. So this is a cross section, like it has been cut like this. So in here you can clearly see that it has not infiltrate to the nerve. Instead, it has pushed the nerve away eccentrically. So the size of this can be variable. It can be from centimeters to large masses. Also, they are seen solitary in the places. So, when discussing about the macroscopic feature of the schwannoma, it is important to know about the cut surface. So, this cut surface is firm and rubbery in consistency. And as you can see, it is gray, white, and ash in color. And it may show cystic degenerations or these hemorrhages. Hemorrhages are not prominent because this is a benign tumor. So also you can see calcifications occasionally. So you have to remember these things when you are describing about the macroscopic features. Uh, 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 features apart from these things. So now we are going to talk about the neurofibroma. So neurofibroma is a benign peripheral nerve sheath tumor which is composed of mixture of Schwann cells, perineural cells, and fibroblasts, and also axons. So fibroma is a mixture of Schwann cells, perineural cells, fibroblasts, and axons. Unlike schwannoma, which has pure Schwann cells. Also, this is not encapsulated and this infiltrates the nerve, which is also not seen in schwannoma. So, what about the etiology and genetics? So, this is a sporadic associated with neurofibromatosis type 1. The schwannoma was associated with type 2 and this is associated with type 1. So, the mutation of NF1 gene or the chromosome 17 causes the reduction of neurofibrin. This will increase the RAS activity and it will cause the tumor growth. So let us elaborate this a little bit more so you can understand clearly. So why is RAS important? RAS is a proto-oncogene signaling protein. So when this is activated, it triggers MAPK pathway cell proliferation survival signals. Normally, RAS is switched on temporarily, but it get off although it is temporarily on. But in activation of NF1, RAS is always on. So it causes uncontrolled growth because of these things, because of these triggers. So there are mainly three types of neurofibroma, the localized one, diffused one, and the plexiform one. Localized one is the most commonly seen one and it is solitary and cutaneous. And the most important thing one is the plexiform because it can undergo malignant transformation. When we're discussing about the gross features of neurofibroma, as you can see here, they are well circumscribed and when touching them, they feel like the consistency is firm and rubbery and you can see some cystic lesions also there. And also, as I said earlier, it is not encapsulated, and it rarely undergo the malignant transformation, as I said earlier. And also, it infiltrates into the nerve. Look at this thing. This is the one we discussed earlier, and this is the neurofibroma. You can see that it goes with the nerve, not separately. It goes with the nerve. So the axons can be seen in this neurofibroma. So microscopic features, there are spindle cells with wavy buccal nuclei. It means there are pointed these like nuclei. I can show them to you. So look at these nuclei. Sorry. Look at these nucleus. These nucleus, they are wavy buccal shaped nucleus which are pointed. And also the background has a loose myxoid stroma. The background is loose myxoid stroma. You see as white color things here. And collagen shredded carrot appearance is there, and to identify this is mostly helpful about because there are high amount of mast cells, and also there are axons present within the tumor, it is the most key difference from the schwannoma. So when we discuss about neurofibroma and schwannoma by comparing, uh, there is no capsule in fibroma, but there is a capsule in schwannoma. Neurofibroma, there is no capsule, and schwannoma, there is a capsule. And relationship to the tip to the nerve, fibroma is infiltrates, but in schwannoma, it just pushes the nerve aside. And when talking about the cells, neurofibroma mixed, it is formed by the mixture of Schwann cells, perineural cells, and fibroblasts, and schwannomas are formed purely from the Schwann cells. And regarding the axons, neurofibromas contain axons, but Schwann cells does not contain axons. Well, in histological way, the neurofibroma has wavy nuclei and myxoid stroma, while schwannomas has Anthony AB areas and Verroque bodies. So those are the things that mainly discussed in here, and in the next video we are going to discuss about the adipose tissue tumors.